Hey, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 121st seminar, and I call it a DTU Denmark Day. I am in a hotel room in Indiana where I am visiting to give an invited talk at Indiana University. Unfortunately, I missed the most amazing sky show in a so-called solar eclipse by a couple of days because I just got here yesterday. And of course, my last name is Moon, and I blocked the sun, I guess, but I, visit this amazing, Indiana, because at the time, Indiana, enjoyed, amazing total eclipse, basically making entire world black. The Indiana area only black. And then I heard even animals stop making sound or the singing. And then that was an amazing event, at the time, I believe. Right after this trip, I will go to Toulouse, France, to attend and give a talk at the SynBYSS conference. If you are attending the conference, let's catch up there. Okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do so, but I will try, our pioneer speaker, Professor Pablo Nikel. He doesn't need any introduction because we all know him. He is a professor at DTU, Denmark. That's why I call today DTU Denmark Day. I believe we connected first via social media like LinkedIn before meeting in person last year at, I guess, the 2023 SynBYSS conference. That's actually I'm going to visit next week. But at the time was in this Minnesota. And then again in Singapore, where I organized Metabolic Engineering 15 with Matthew Chang and others. He's now helping me again to organize the inaugural Synthetic Biology Conference, SynBYSS conference in Hawaii. He's a world leader in metabolic engineering and the chief editor of Current Opinion in Biotechnology. And that means, if you wanted to submit something to that journal, he is very powerful person. So probably connecting with him, listening to his vision might be helpful to shape your next review article in that high impact, journal. Pablo, thank you so much for your scientific contribution and service to the community as the chief editor, and the virtual podium is all yours now. And thank you again.
Thank you very much for the introduction. So I will just start by sharing the slides. I just prepare a few of them to get introduced. And well, I wanted to say thank you for the opportunity to be part of this tremendously impactful initiative that TAISEC has organized and is following over the years. This is really important for the community, and I'm very honored to be here today. As you may have seen, I call this very brief introduction of what I envision for the near future of synthetic biology and metabolic engineering, rewriting the periodic table of life. But before we go into that, as TAISEC mentioned, we are located in Denmark, in the Copenhagen area. This is the Center for Biosustainability, which looks like that. We are essentially a little bit north to the capital city of Denmark. And this is the team that I have the pleasure to lead there in DTU Biosustain, the Systems Environmental Microbiology team, who are actually the heroes behind the things that we do. So in here you have a very brief introduction of the things that we are interested in, but just to capture that in one or two sentences, we are interested in taming environmental bacteria, alternative cell factories beyond the model organisms to run chemistries that are usually not part of life. We want to engineer microbes to run chemistries, to produce chemicals, to produce polymers that are usually beyond the scope of natural chemistry. So we want to create a new biochemistry. And I think you will all agree that if we have a look at how how any cell looks like in terms of the biochemistry, we can essentially slice it and see an existing biochemistry, which is just a fraction of all the reactions that could happen inside a cell. So what we want to do is to create metabolism to explore this boundary between what is there, the reactions we can see, and the reactions that could be potentially engineered in a living cell, which is this space of allowed bioreactions. And more to the point, this, what you may call cis chemistry, is built on a very limited number of chemical elements from the periodic table. So what we want to do is to create a neo-chemistry, a novel chemistry, whereby we can start using new building blocks. And we are particularly interested in blending in halogen atoms. And the idea goes a little bit beyond what is the state of the art in synthetic biology, which is essentially recoding genomes, right? We know how to do that. We know how to change bases in the genome. We know how to modify the information encoded in a microbial genome. What we propose is to rewrite and recode biochemistry using synthetic metabolism. And the idea here is that we need new building blocks, and that's essentially new atoms. If we are able to do this, in the future, we will be able to produce essentially any molecule that we want by bringing these new chemistries into life. This is also, goes hand in hand with discovering new enzymes, and where there isn't any enzyme that can run the chemistries we want, creating new enzymes and then convincing the cells to run these new pathways, these new reactions, and producing these compounds that are usually not part of the chemistry of the cells. So that, in a nutshell, is what we are doing in my team in DTU Biosustain. Happy to discuss more details with whoever might be interested. Reach out. This talk is being recorded, but in the first slide, you have my contact details. So please feel free just to go ahead and get in contact. And again, thank you very much, Taisuk, for this opportunity to share a little bit of what we are doing here, and looking forward to be in contact with the broad community. Thank you.
Pablo, amazing. It's my great honor to have you here. So thank you so much. This is amazing talk. Also, I visited DTU, I think 2022. Unfortunately, I missed you at the time, but amazing institute, amazing scientists and engineers. So that is probably one of the best, university in the world, And also, I think the next year, the metabolic engineering conference will happening in that university, near the university, I believe. So that is another amazing, opportunity for us to visit again DTU and then interact.
Absolutely. I'll be co-chairing the Metabolic Engineering Conference along with Irina Borodina and other people as well. So you are all invited to Copenhagen in June 25.
Yes. I'm so excited to visit again. So I hope to connect. And also, if the Novo foundation involved, there's always great party, I would say. All the great food, drink, and
great food we can ensure. In June, we can almost ensure good weather. I wouldn't say fantastic, but at least good weather. So we are looking very much forward to receive the whole community again after Singapore in Copenhagen.
And also the night will be starting 11 p.m. I guess, so we just keep drinking until dark, right?
Exactly.
And with the light. That's the very amazing thing. All right, thank you so much.
Thank you very much.
Hey, and interesting, yesterday I talked about, the chemistry you just described with the colleague here. So that's amazing, opportunity for us to explore in the future. So thank you for leading, pioneering those kind of field. So we are absolutely grateful what you have done. Thank you.
Thank you. Thank you.
All right. So now the main speaker of today with a longer introduction, Dr. Quentin Dudley is an assistant professor. I heard just started this year, January. So congrats, your new achievement and the exciting moment. And he's now in chemical and biological engineering department at the University of Wisconsin-Madison, where his lab will build tools for cell-free and plant-based bioproduction of molecules. It's kind of similar to what Pablo has been doing. He completed his PhD in the lab of Michael Jewett at Northwestern University, where he developed an in vitro prototyping platform for assembling and optimizing multi-step enzyme pathway. That's very interesting because that's another topic we discussed yesterday with chemists here. As a postdoc at Alham Institute and Joninles Center in Norwich, UK, under joint mentorship of Nicola Patron and Sarah O'Connor, he used CRISPR-Cas9-based genome engineering to improve the wild tobacco as a chassis for the bioproduction of medicinal protein and metabolite. His recent experience include time at the synthetic biology startup, Calyxt, which is engineering bioreactor-based plant cell to produce cosmetic, nutraceutical, and pharmaceutical ingredients and speculative technologies. A non-profit, the speculative technology, a non-profit that runs coordinated research programs to unlock materials and manufacturing technology that don't have a home in other institutes. Interestingly, Sarah O'Connor, now in Germany, I visited her, I think 2 years ago, and then giving seminar. And also she is an absolutely amazing scientist. And she used to be also my committee member at MIT. And I guess this is kind of a connection between him and me. It's kind of supervised by one of the amazing scientists in the world, and also Sarah also gave a talk for this seminar series. Quentin, thanks so much for your time today. Please take it away. It's all yours now. Thank you.
Okay, let me get this screen set up.
Yeah, your slide looking perfect.
Yep, you can see me okay?
Yeah, the slide looks perfect, yes. But your voice a little bit, small, so can you probably shout?
Yep. Hopefully this is clear. Can you hear me okay?
Perfect. Go ahead.
Great. Well, thanks so much for that kind introduction, Tae-Suk. And yeah, what I'm going to tell you today is a little bit about my past research and how that's going to set up my research group at the University of Wisconsin, where we're going to be focusing on plant and cell-free synthetic biology for sustainable chemical production. So showing here is picture of the campus at Madison, looking down the mall towards the engineering building. You can see the Wisconsin Badgers Stadium at Camp Randall, and if you've never been to Madison, it's a beautiful city of lakes, and would encourage you to visit at some time. So I've been interested for a long time in the ideals of metabolic engineering, that we can harness biology to generate sustainable chemicals, very much how Pablo was describing earlier. And we can use biology then to make chemical transformations at ambient temperature and pressure to create low value things like biofuels, medium value things like commodity chemical precursors, polymers or high-value pharmaceuticals. And so Tae-Suk already really covered kind of my introduction, but this interest for me started in my PhD, again at Northwestern, where I got into how bacteria can do these transformations and what options we have to take the crude lysates of these cells and still activate complex metabolism. But I wanted to kind of shift in my postdoc to what something that felt a little more like the cutting edge or the new area of synthetic biology. And that's what inspired me to do my postdoc abroad in the United Kingdom. And so today's talk is really going to be a distillation of my postdoc work. But I also really value my time in industry. I came back to work at Calyxt and actually see many of the challenges that I was experiencing in my postdoc about limited tools for engineering plant metabolism playing out in real life. And that gave me a lot of confidence as I was working for this science funding agency to again start my own group here at Wisconsin, and now just a few months into that process. So we'll get started here. And what I'm going to mainly talk about is my postdoc work, which is going to be reconstituting monoterpene indole alkaloid biosynthesis in a genome engineered tobacco plant. And so we'll start with that, and then at the end, I'll talk about how that work sets up what I'd like to do here at Wisconsin. So again, my postdoc was in this small city north and east of London, here in Norwich. And I was in a really unique opportunity to have two really solid mentors on this project. Nicola Patron at the Earlham Institute has recently moved to University of Cambridge, really an expert in synthetic biology, as well as Sarah O'Connor, who at the time was at the John Innes Center in Norwich, and as Tae-Suk mentioned, a real leader in specifically alkaloid biosynthesis and pathway discovery. And so this project was really a merger of their backgrounds to try and engineer this metabolism practically. And so the target molecule that inspired this project was this cancer therapeutic called vinblastine. It's been known for many years, since the 60s, as a potent microtubule inhibitor, and has proved a frontline treatment for Hodgkin's lymphoma and other blood cancers, particularly in children. And this is a natural product made in plants. And so the natural source of this plant, or excuse me, of this compound is this Catharanthus roseus plant, commonly known as the Madagascar periwinkle. And it produces vinblastine as well as 130 other compounds in this chemical class called monoterpene indole alkaloids. But the challenge is that these compounds accumulate at extremely low levels in this plant, making collection, harvesting, and accumulation of these molecules still laborious and expensive. So the metabolic pathway to vinblastine is incredibly complex. I would argue one of the most complex pathways that science has elucidated to date. And this was 29 unique metabolic reactions starting from central intermediates tryptophan, as well as the isoprenoid pathway producing this 10-carbon precursor. So you have the terpene part of a monoterpene indole alkaloid coming here, and the alkaloid part coming from the nitrogen in this tryptophan. And so the last steps to be discovered was this region right here, which was done in Sarah's group in 2018 with similar work coming out of the DeLuca lab around that same time. And that was really exciting that now we're finally able to have the full pathway to this molecule. But we wanted to engineer it outside of this plant because Catharanthus roseus is notoriously difficult to work with. It's very difficult to transform and manipulate genomically. So we wanted to reconstitute this pathway in a heterologous host. And a standard choice would be to use, for example, a yeast cell. And there had been efforts in the O'Connor lab to do this, but they proved challenging. And those initial efforts were focused on making this intermediate compound strictosine. And so strictosine is about halfway through the vinblastine pathway, and it really is the branch point for these monoterpene indole alkaloids. And so that's where you get about 3,000 different molecules have been identified across many plant species. And here's a list of some of the more interesting ones. And you might even recognize a few of them. So strychnine and quinine, known for their medicinal properties for many years. More recently, ibogaine has gotten interest as an anti-addictive treatment, and as well as several other compounds for treatments for pain, cancer, and other areas. So so that's where my focus in this project was to produce strictosine. And the O'Connor Lab's efforts in this space prior to me starting had attempted to do this in yeast, but we found that to be challenging. Even knowing the rate limiting step, this first hydroxylation, the previous efforts could not get particularly high titers of strictosine. And so our goal was, rather than use yeast, we wanted to reconstitute this pathway in something more phylogenetically similar to the Madagascar periwinkle, the wild tobacco Nicotiana benthamiana. And that's the focus of my project and talk here. So why Nicotiana benthamiana? Let me introduce this plant to you. And so it is a wild relative of tobacco. Nicotiana tabacum would be the plant you're probably more familiar with. And this wild relative of tobacco has, for a long time, been a model species for plant microbe interactions. It's native to Australia, and during its evolutionary trajectory, it developed a few mutations in its immune system that means it has honestly a broken immune system. That's what makes it a really good host or chassis or model species for studying interactions with pathogens. But notably, that broken immune systems enables this phenomena of high-level transient expression. So what you can do is take a gene of interest and put it here in a shuttle bacterium. This is a native soil bacterium that infects many plants in addition to tobacco. And it's been domesticated to remove its virulence genes, and those virulence genes have been replaced with a gene of interest, depicted in this gray coding region here. And you can actually just syringe the liquid, the growth media containing Agrobacterium into the leaf, and then just 5 days later see substantial overexpression of a protein. Or you can use this technique to downregulate or silence native genes in the tobacco. And so it's a really useful tool for basic science. But this phenomenon of transient expression has also enabled us to reconstitute metabolic pathways. And that's because if we want to overexpress, for example, a 5-step pathway, we can take each of those 5 enzymes, put them separately in a different strain of Agrobacterium, mix those strains together, infiltrate them, and get all 5 genes overexpressed at the same time. And the limit is not just 5, but even 15 to 20 enzymes. So the pathway to strictosine that I'm going to talk about today required 15 separate enzymes or proteins to be expressed at the same time. It's a really unique workflow. And this approach has enabled a wide variety of different metabolic pathways. It's really started to become the gold standard for helping discover these biosynthetic pathways. And I'm showing an incomplete list of all the different compounds that have been made in this space. I'll just highlight some recent work discovering this QS21 vaccine adjuvant, and a lot of activity in the last few months on discovering the biosynthetic pathway to the cancer treatment drug Taxol. So not only can you put small amounts of agrobacterium into tobacco and get just peaks on a LCMS trace, Benthamiana is also a scalable bioproduction platform. And that's because you can, rather than using a hand syringe, you can put your gene of interest in agro, and then to get all of your leaf tissue to express your gene of interest, you can flip it upside down and dunk it in a bath of agrobacterium growing here. And then do that transient overexpression. You can see that all the leaf tissue is expressing GFP in this case, and the parts that aren't are only just the bits of leaf that have grown in the last 5 days. And so this has meant people are starting to use Benthamiana to produce not just metabolites, but also therapeutic proteins. And so you can imagine greenhouses filled with little plants, do your infiltration, and then harvesting the leaf material and then isolating a unique protein. And examples of this from the therapeutic side have included monoclonal antibodies for treatment of Ebola, as well as virus-like particles, which can be used as vaccines for the flu, for Zika, and polio. And there was a successful COVID-19 vaccine made and approved by Canada in 2022. So a really exciting host and chassis organism for metabolic engineering in plants. The challenge that I ran into as I was starting to work with the pathway to streptocidine is that Benthamiana is great in that it provides a lot of the precursors that you need for the metabolic pathways that I shown on that slide. It's a folded, it is able to express and fold your proteins. There's often helper chaperone proteins that enable folding or turnover of cofactors. But one of the challenges is that there's all this other metabolism happening in the plant. And what we observed as the previous efforts in this pathway by this Nature Communications paper in 2014, as well as other this group in 2016 and the postdoc that worked on this project before me, is that we saw derivatives of these pathway intermediates accumulating in the plant. What do we mean by derivatives? This would be where the intermediate is getting modified, for example, by the addition of glutathione here. We saw where we would get over-oxidation of these molecules. We also show substantial unwanted glycosylation, shown in red, as well as acylation. And so we were this is observed here, where you see this LCMS trace. This is with the empty vector, and here just expressing the genes for the isoprenoid pathway, as well as this geraniol synthase to produce geraniol. You start to see all of these other peaks showing up. For example, this hexosyl hydroxygeraniol, or with 2 hexose groups, and here's an example of over-oxidation. And so this prompted my project to have 3 strategies to enable production in Benthamiana. First, we thought, how can we knock out potentially these metabolic reactions that we don't want? So that would be approach 1. And the analogy, if you imagine metabolic flux as a pipe, where you want more of your flux, your liquid, to go towards your product, this would be how we're going to fix the leaky pipes. I'm also going to briefly talk about a second approach where we're going to remove a co-purifying metabolite. In the analogy, that would be turning off a polluting pipe. And in approach 3, we're going to increase our accumulation of our target compound by widening the pipe, by adding an additional enzyme that completes the pathway. So starting with approach 1 was this knockout strategy. How can we get rid of this unwanted derivatization? And so by looking at the different types of modifications we see, we can hypothesize what types of enzymes do these reactions. For example, glutathione transferases, different cytochrome P450s or oxidases, glycosyl transferases, and acyl transferases. But it wasn't clear there's how many of these candidate enzymes were in our plant, and how should we select which candidates for knockout. We used 4 different approaches. First, we had to identify exactly which enzymes were in our plant, and then we used 3 techniques of transcriptomics, phylogenetic comparison, and in vitro activity screening. And I'll walk through each of those here briefly. So first, we needed to know what are the sequences of these enzymes? And when I started this project in 2017, the existing draft genomes were highly fragmented. So there's 2 genomes available based on short read sequencing. And again, it wasn't clear what the actual sequences were. Are there chimeras of related genes? And different misassemblies were possible. So we actually initiated an in-house effort at the Earlham Institute to resequence this plant and combine short reads from Illumina along with bar-coded reads from 10x Genomics and long reads from Oxford Nanopore to create a much higher fidelity assembly. And this became a little toy project for the folks at Earlham Institute to test some assembly algorithms. And ultimately, we came out with our improved genome along with several other groups all around the same time in 2022, 2023. So this was really helpful, and that now we can be much more specific about how many candidate genes were actually under consideration. But how do we down select this list? There are still far too many for us to make knockouts of all of these in a plant with the technologies available to us. So we started with the transcriptomics approach, hypothesizing that maybe when these compounds accumulate, the plant has a dynamic response to overexpress different derivatization enzymes in response. So we had 4 different conditions, 2 of which accumulated these molecules. And we looked for different profiles of genes. And we were particularly interested in some of these, that when you have these geraniol and the pedolactyl intermediates accumulating, you see higher counts of this glutathione transferase. Another approach we used was phylogenetic comparison. So here we're looking at a phylogenetic tree of cytochrome P450s. These 4 marked sequences are known to be active on the strictosine biosynthesis pathway. So we chose enzymes from related clades. And we did the similar approach for glycosyltransferases, where each of these sequences marked with a diamond or a star or a triangle had shown in vitro activity on geraniol or related compounds. So that was the second strategy we used. And then finally, I wanted to build on my postdoc, or excuse me, my PhD work, which was using cell-free lysates to express proteins. And so this approach involves taking a growing organism, lysing those cells, and producing this lysate or extract here. And in my case, I was using E. coli to source the lysate, and then adding in substrate amino acids for your protein, substrate NTPs to do your transcription reaction and make your RNA, as well as a number of small molecules to make ATP, and then a DNA template. And this has worked really well for producing things like GFP. And during my postdoc, I adapted this effort to express some plant proteins as well. And we were right next door to the Earlham Biofoundry. And so we were able to put this workflow in 384 well plates and assemble these reactions using acoustic liquid handling using this echo device, where you take a 384 well plate, transduce sound through the bottom, droplets fly up into a second 384 well plate, and then you have the ability to assemble different reactions with different conditions. And we showed that by swapping out different N-terminal expression tags, so that's where we're showing different tags on this axis, different sequences of UDP glucose-dependent glycosyltransferases, and showing that different tags did indeed help improve expression of several of these plants, and showed that the initial skeleton of a pipeline for then screening those enzymes and showing that new peaks appear in the presence of some of the pathway intermediates. So the upshot of all these strategies is we were then able to identify 34 gene candidates of interest, mostly glycosyltransferases and a few glutathione transferases and cytochrome P450s. But then we had to actually go knock those out. And that is a rather laborious process in plants, at least when I first started this project, where we wanted to use the well-described CRISPR-Cas9 system, where we're going to introduce a double-stranded break guided by this sgRNA. This 20 nucleotide sequence directs the nuclease where to cut. Once it makes that cut, random deletions or insertions will accumulate in the germline plants, leading to gene inactivation. And so the state of the art when we started this was to deliver a construct encoding Cas9, a selectable marker, and then your different guides here represented in green. And you deliver that to a plant using tissue culture, where you grow plant cells in this callus form, then add different plant hormones to induce root formation, several more hormones later then to introduce shoot formation, and finally get a viable plant out the end. And this tissue culture takes a long time. It's 3 months of weekly subculturing and 6 to 8 months to get that first, the next generation of seed. And so the efforts done in this project were done by Wendy Harwood's group at the John Innes Center. Incredibly helpful to us. But halfway through my postdoc, a new technique for genome engineering tobacco came out. This was developed by Dan Voitis's group at the University of Minnesota, and that involved delivering the guide RNA to a benthamiana plant that is already expressing the Cas9. And it's expressing that Cas9 stably, but nothing's happening because there's no guides. But if you deliver the guides then with a viral vector, that's this tobacco rattle virus genome encoded here, you can then get expression of your guides. And because they have this little mobile element, this epitope of RNA attached, they can move through the plant vasculature to the meristem, where then a genome edit occurs, and then all the plant tissue that grows above that point of editing is going to be mutated. So you can get good you can get stably mutated plants with just a single infiltration, so much less work and shorter time. And so even though this came out rather late in this postdoc arc, over half my edits were made with this approach. And so with those 34 candidate genes, we did a lot of plant genotyping and ultimately tested 18 T1 lines with stable mutations. And here's the list of several of the UGTs that we knocked out. So we then screened these mutant plants and identified 2 glycosyltransferases of interest. And let me show you how we screened these. First, I made putative identifications of 20 different peaks that accumulated when we expressed different pathway enzymes towards strictosine here. And then we tracked all 20 of those peaks and measured them across, in this case, I'm showing data for 11 tobacco lines with knockouts in these glycosyltransferases. And you can see the different, for peak 1, here's the counts. And interestingly, for this knockout of this UGT, we see loss of these peaks in these 3 positions. And we also see mutants in this 85A73 sequence also lose peaks. So that in many ways did validate our hypothesis that by making mutations, we could remove some of these unwanted metabolites. And so that was kind of the arc of approach 1. Approach 2 was then to remove a co-purifying contaminant metabolite. And if you're thinking of tobacco, what is one of the first contaminant metabolites that might come to mind? It was nicotine, was something we wanted to remove. And this was a collaborative project, considering it's Taesuk highlighted it's the Denmark Day. This was a collaboration with the Guy Flores lab at the University of Copenhagen. And so they were interested in this metabolic pathway and wanting to make a nicotine-free tobacco. And so here's the biosynthetic pathway to nicotine, which we wanted to make mutations that would minimize the impact on the plant. And so the hypothesis is that we would target for knockout just these last steps, this last hypothesized step of the pathway, this berberine bridge-like enzyme. And so we made, so the idea was to knock out this enzyme. And so we initially identified 3 sequences for knockout and made some triple knockouts. Then with our improved genome assembly, we found a 4th sequence and then a truncated version of that. So made guides for all of these, and then ultimately made several triple and a quintuple knockout, and validated the hypothesis, at least for annotating. We have our wild type sequences here, and then the knockouts showing no accumulation. But for anabasine and nicotine, we reduced our levels of nicotine, but we didn't knock them out all the way. That was a little disappointing. But to kind of follow up this story, a lot of work in just one slide, Katharina and others in the Guy Flores lab actually did some further analysis on these lines with the nicotine here. And most of the nicotine that accumulates in tobacco species is S-nicotine. And so that's what's here. But they noted that what was accumulating in these plants was actually a racemic mixture of both S-nicotine and R-nicotine. And so that actually gave us insight to the biosynthetic pathway of how these steps are working. And we hypothesized that actually this accumulation is from spontaneous ring coupling, which leads to the racemic process. And so Fernando's group now has some really nice, we put a nice new phytologist paper out hypothesizing how 2 different possible routes and how BBL could be involved in nicotine biosynthesis. So finally, the step that ultimately enabled strictosine production in a plant was this approach 3, completing the pathway with an extra enzyme. And so the previous efforts in this space by Meaton et al. Had been able to produce this 7-DLA molecule here, but they hadn't been able to put all 15 genes into one plant. They were able to feed an intermediate here and get to strictosine, but not the whole bit. Where parallel work in the O'Connor lab was actually working on this species of plant, Nepeta. And Nepeta produces these molecules that this is what known as catmint or catnip that gives this euphoric effect on cats is due to these iridoid molecules. And the biosynthesis pathway actually shares some overlap with the monoterpene indolachloride pathway here. And so work by Benji Lichman actually identified a key enzyme here called MLPL, which not only orients the stereospecificity of this ring closure, but accelerates its rate substantially. And so our hypothesis was that by adding this MLPL enzyme to our pathway, that might help strictosine production. And that's what happened. So the next slide, I'm going to show the production of 7-DLA with and without MLPL. So we have our empty vector here. We have production of geraniol. Here's then our whole pathway needed to make 7-DLA. Doesn't make any peaks that match up with this retention time. But when we do add this MLPL, we see a much larger peak at this correct retention time for 7-DLA. So adding MLPL did turn out to be a secret sauce intervention for producing strictosine. So with our ability to make 7-DLA in hand, I then put the biosynthetic pathway to make 7-DLA here. So these pluses indicate about 10 overexpressed genes. And then we were able to add the final enzymes one at a time to make strictosine. And we see that you add another enzyme here, we make loganic acid. Add the LAMT enzyme and we make loganin, the SLS, and then finally the 2 enzymes to bring in tryptophan, and we're seeing strictosine in our plant without any other metabolites needing to be infiltrated. And so here's how that peak matches up with the strictosine standard, and our final titer at this time was 200 milligrams per kilogram dry weight. So that's really kind of how we were successful in this postdoc project. And just to kind of then give you an update of some of the work that's happened since this project ended, that across different organisms, I'm going to depict the full cartoon of the pathway to vinblastin here. that here's the work that I talked about here, producing streptosine in yellow here in plants, where there's also been some interesting work using cell-free enzyme cascades to make this early intermediate napetolactone. But that MLPL was actually key to build and improve the levels of streptosine production done by the Tang lab at UCLA, and then a real tour de force effort by Jay Kiesling's lab to get these two precursors to vinblastin to accumulate in small levels in yeast. So exciting developments in the monoterpene indole alkaloid space. So to summarize, I've talked about 3 steps for increasing streptosine production, this genome engineering approach, what we learned about nicotine biosynthesis, and then finally the importance of this MLPL enzyme. And you might have a question, well, what happens if you combine approach 3 and 1? And I was really optimistic about this experiment, but unfortunately, when we tried expressing this MLP pathway in our knockout lines, here's the using a wild type plant in dark gray, we actually saw very similar yields of streptosine in our mutant plants, which leads us to think that, you know, we knocked out some of the derivatization enzymes, but there might be more needing to be knocked out for that approach to yield dividends on the back end of a metabolic pathway. So that's most of the science that I wanted to present today. But I wanted to briefly then, with my last few minutes, show how this is going to set up work at the Dudley lab. And so one of the project areas that I want to continue working on is in this, how can we make Nicotiana benthammiana a better platform for expressing these pathways, and also really starting to build on the tools we need for plant synthetic biology. For example, better tools for building gene circuits and toggle switches. And I'm also interested in using benthammiana and other approaches for pathway discovery, potentially in the alkaloid or flavonoid biosynthetic pathways. And that discovery effort can actually be enhanced then, I'm hoping, by project area 2, where we can use these cell-free systems that I briefly alluded to. And didn't have time to go in more on how I use these in my PhD to reconstitute metabolism. But because of their ability to rapidly express proteins and their ability to automate some of these workflows, I'm hoping they can increase our throughput in bioprospecting. And also, if we find enzymes that need metabolic or directed evolution strategies, we can do those more rationally in this cell-free context. And so that could lead to beneficial experiments for plant synthetic biology, potentially, as I mentioned, in enzyme discovery and being able to test hundreds of candidate enzymes instead of just dozens. It could guide genome engineering, as I kind of described here. It could also be really useful in understanding transcriptional networks for regulation in plants, which are often really complex. And so by being able to express and validate transcription factors and what DNA they bind to, cell-free systems could really help in this space. And finally, I talked about microorganisms like yeast as a production platform, much more on plants. But I'm also interested in the long run, how can we just use enzymes in a more of a flow or chemical engineering context without the living cell around? And so this is an area that I'm really excited about and built a roadmap for as I was working for the Speculative Technologies, a nonprofit science funding agency. So really interested to see what future directions my lab could go more in the decade scale timeline. So many of the techniques that the Dudley lab will use, you've already heard about, but particularly wanted to highlight liquid handling automation, high resolution mass spec being coupled here, and opportunities for linking this with artificial intelligence and machine learning for pathway prediction, protein engineering, and optimizing biosynthetic pathways. And just want to then put a plug for plants as a really unique production chassis using sunlight and CO2 to make useful molecules. And so I see plant synthetic biology as this new frontier for biological manufacturing, with the potential to accelerate a transition to a distributed, sustainable, and post-fossil carbon society. And some of the real exciting areas in this space recently, I'm listing here. Our ability to build synthetic metabolic routes to increase plant biomass, putting in new to nature carbon fixation pathways, adding nitrogen fixation to plants that don't natively do that, making plants that respond to environmental cues, potentially by making agrochemicals. And then again, a big highlight of this talk would be the discovery and production of novel medicines. So my final slide is then an acknowledgement of all of the postdoc work that I described. The science here was really a credit to Nicola and Sarah's efforts and their respective research groups, and the funding for that project. And then, yeah, this is one of my first chances to put a picture of the Dudley lab. We've been at research for just a few months here. This is our shiny new homogenizer for making cell lysates. We have one graduate student, several undergrads, and I'm actively soliciting postdocs. So if any of those project areas sounded exciting to you, please reach out via email. And so with that, I'll use whatever time we have remaining for questions.
Fantastic. Thank you so much. You know what? You have very unique expertise combining plants, synthetic biology, and cell-free system. So, you know, I wanted to say, if you are listening to this talk and then wanted to work with a great advisor, just apply and send email to him, and you will be, you know, successfully, you know, achieving a lot of great things with him. So thank you so much again. Also, a very interesting thing I want to say is, you know, I'm very much supporting plant synthetic biology, even though I'm not doing plant synthetic biology. That's the reason I recently recruit and hire one of the associate editor for my journal, New Biotechnology. And she, you know, I cannot tell you now, but she will be associate editor focusing on plant synthetic biology. So because this is a very important field, I will say, so I'm gonna emphasize how important and then encourage more people working on plant synthetic biology. And so absolutely brilliant talk. Thank you again. Let me see whether I have any question from audience. Let me start some, you know, question and comment. So I'm also excited you also, you know, one of the experts in the, you know, cell-free system. Actually, one of the main theme of the conference I'm attending next week is cell-free, basically, you know, very focused cell-free system, conference, because this is the build a cell conference, and then one of the main topics cell-free. The question I want to ask is, I see, you know, huge potential in the cell-free system in terms of prototyping, as you kind of briefly touch about that one. And then, but at the end of the day, you know, we need to deliver the real product in the market. I see a lot of, you know, great, you know, achievement, including, you know, diagnostic cell-free system, you know, by a lot of, you know, amazing leaders in this field for the disease detection, etc. Prototyping is one of the great avenue for cell-free. But in terms of the large scale, you know, production, you know, already we knew, I mean, from the vaccine, COVID vaccine production, so we already demonstrate some. But to make that one step further, I want to make somebody, probably not gonna be somebody, including you, making some large scale production of not only high value chemical, like, you know, pharmaceuticals, but small, you know, bulk chemical, just using cell-free system rather than, you know, host cellular system. But still, challenging would be, you know, economics, I guess. Could you kind of on that aspect? What kind of challenge we have? You probably touch a little bit, but I want to hear your more in-depth kind of analysis or insight.
Yeah, no, I think in principle, you know, broadly, I'll just say kind of people think of cell-free systems in kind of two bins. One is production of proteins, and one is production of metabolic pathways or small molecules. So you can either use these, have the full transcription translation apparatus to make a protein, or just get your enzyme. You can make that with your cell-free system, or you can make it conventionally and isolate it, and then daisy chain these metabolic reactions together by either immobilizing these enzymes or putting them in batch formats. And so I'm going to choose to answer. So I guess I'll answer for the protein side. I'm seeing some interest in scenarios where you rapidly need to change the protein that you're making. And so I think some of the areas where you're doing personalized medicine and making different protein therapeutics on demand, that's where there's been, I think that'll be some of the killer app areas for lack of a better term. And I think that'll be a nice complementary technology to some of the mRNA vaccines, but things that need protein therapeutics. But I know maybe a little less about that area. What I am really excited about is the second option I mentioned, these enzyme cascades. And again, we're doing large scale biotransformations with enzymes for processing corn for ethanol production, for cellulosic biofuels, for there's been a lot of interest in pharmaceuticals for using enzymes. The reasons that I don't think they're more widely used are the cost metrics that you mentioned. And it's generally that the enzymes, you have a fixed cost to produce them. And it really depends on how much longevity and how many turnover events you get from an enzyme. So, glucose isomerase famously has a half-life of almost a year for maize corn processing, but most enzymes are a day or most.
Got it.
So what got me really excited as I was working on this speculative technologies roadmap is looking at this exciting space of protein engineering. And there's so much effort with machine learning, with de novo design of proteins, with directed evolution. I think we're going to see so much advancement here that one of the metrics we can put in is we want these enzymes to be really robust, to last for weeks at a time. And I think as we start to get to those types of proteins, more products will be amenable to cell-free. Small molecules, at least, will be amenable to cell-free synthesis. The other area that I think is going to be a challenge is, reactions often don't happen in isolation.
Sure.
A to B to C. You need cofactors. You need other molecules, particularly ATP and NADP. And so systems for recycling those potentially directly from, in the case of electron carriers like NADH, systems to directly regenerate those from electricity using electrochemistry. I'm really excited and bullish about the opportunities in that space because cofactors are kind of your second big cost in these cell-free systems.
Got it. Thank you so much. That's very interesting because as I mentioned earlier, I met chemists here, also have multiple meetings, also with the chemists, we have the dinner, amazing dinner together. The discussion we had is basically you summarize somehow, for expert kind of perspective, because they said they are chemists, but not that many chemistry department doing so much, but nowadays more, but we want to see more, in the chemistry department. So that was kind of very interesting discussion for me. That's why I'm asking that question to you. I see some question from audience. Maya ask, since you talk about cell-free plant system, some systems have focused on entirely reconstituted system pure. Do reconstituted plant transcription translation system have inherent value, or is the value in the variety of chaperone you find in the lysate? I try to understand the question.
Yeah, I think maybe I'll try and rephrase the question is there's a number of different cell-free expression platforms out there. The question asker was talking about this so-called pure system where you isolate each of the transcription translational components, everything's purified, or you can use these crude lysate approaches where you make you isolate a crude lysate either from E. coli or for other organisms. There do exist expression systems derived from plants, most notably from wheat germ. And then there's a more recent set of protocols using Nicotiana tabacum suspension cells. And then there's a handful of other papers, but wheat germ and tobacco are two of the leading candidates. And I think it's going to depend the type of molecule that you want to make. And if it works in some of these prokaryotic systems, those often have lower costs to produce and higher yields. And so if those work, great. Some of my unpublished data is that's a question that's very interesting to me. So far, some of the limited data I've collected is not all plant proteins work or express and fold in these prokaryotic systems where you have a higher fraction that express in a wheat germ kit, for example. But it's not always predictive. Some sequences do work better in the prokaryotic, and so again, chaperones might be really important, the folding environment. And so those are exactly the type of questions that I'm interested in pursuing in the new lab.
Sure. So I mean that's very fascinating because I kind of fascinated by the fact cell-free become very popular, of course, pioneered by, Michael Druid, your advisor, very recently. But I, got to know about cell-free system very early in my career, even that is the 1907, because I work with some, people in Korea, including Dong-myung Kim. He's actually mentor of the Michael Druid many a decades ago, more than decades ago. And Dong-myung Kim is sort of pioneer, with whatever, the cell-free system, etc. But now still, suddenly it become popular because Michael Druid kind of pitch very well to the community, I guess. So that's kind of amazing because to me, that's old technology, but now people think that's the new technology, but that's not the case. Because now we have 11:08 Central Time or afternoon, basically the Eastern Time I'm currently in. We may just cross and then we may do just informal chat. Okay. So thank you again all for joining and staying today. Also the speaker today. We'll meet again on April 18. Thursday, the same time, the same Zoom link. We'll have the Professor Stanley Chi at Stanford University and Dr. Oskar Stauffer at Leibniz Institute for New Materials in Germany. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I'll stop recording. Just give me 1 second.